Thank you for stopping at our virtual poster. We study CA1 pyramidal neurons in the hippocampus. These neurons are place cells that tend to fire in a specific place field. From in vivo experiments from many researchers, we know that the center of mass of the place field shows a backward shift with experience, and that this shift is strongest in a novel environment. We think that ion channels intrinsic to CA1 pyramidal cells may contribute to this shift through modulation by acetylcholine, which is known to be elevated in novel situations. We used in vitro electrophysiology in slices to investigate the responses of CA1 pyramidal cells to depolarizing ramps that simulate the depolarizing input that place cells receive when an animal traverses the place field. We then investigated the modulation of these responses by cholinergic activity. Figure 1 shows the responses when ramps are injected in the soma in A or in the dendrites in B. In all cases, the neurons tend to fire more spikes at faster frequencies on the up ramp than they do on the down ramp, giving rise to asymmetric responses that are shifted earlier in the ramp. The inset plots of instantaneous frequency versus injected current show this asymmetry. We quantified the difference in spiking by plotting the spike ratio in this bar graph. This is the number of spikes on the up ramp divided by the number of spikes on the down ramp. And you can see that this asymmetry is more prevalent in the dendrites than it is in the soma. We want to know what currents underlie this asymmetry. And in figure 2, we looked at potassium currents that are known to mediate adaptation. These are the calcium-activated SK current, the M-type current carried by Kv7 channels, and the A-type current carried by Kv 4.2 channels. When we block any of these currents, we don't see any effect on the asymmetry. Other potassium currents that we investigated were erg and delayed rectifiers carried by Kv1 and Kv2. In figure 3a, you can see that a blocker for the erg current did not change the response. Block of Kv1 did not shift the asymmetry, but it did produce high-frequency doublet firing early in the ramp. Block of Kv2 resulted in burst firing, which is something that other researchers have shown. We then looked at the cholinergic modulation of these responses. In figure 4, middle column traces, we see that carbachol shifts the firing pattern such that the cells now fire more spikes at higher frequencies on the down ramp. Note that the amplitude of the ramps is calibrated to produce similar peak frequencies with and without carbachol. This shift to the right is due at least in part to the activation of the calcium-activated non-selective cationic current, or ICAN, carried by TRP channels. Flufenamic acid, a broad-spectrum blocker of TRP channels, added on top of carbachol, reverses carbachol's effect, and the neurons go back to firing more on the up ramp. We think that TRPM4 channels are largely responsible for this, as their specific blockers, CBA and 9-phenanthrol, which is not shown here, also reverse the effect of cholinergic activation, as you can see here. In figure 4c, you can see that we have some mixed results when we repeat this experiment with SKF 96365, which is a TRPC blocker. So it is unclear at this point to what extent TRPC might also be involved. If TRPM4 is the main effector, that suggests that the cholinergic-induced shift in the firing depends on calcium accumulation, because this channel is activated by increases in intracellular calcium. We tested this hypothesis by chelating intracellular calcium with 10 millimolar BAPTA in figure 5. We were surprised to find that carbachol is still able to shift the firing to the right. One explanation for this is that while TRPM4 is no longer activated, given the lack of intracellular calcium, TRPC may actually be made available by the lack of calcium. TRPC can be held in its closed state by calmodulin, but only when sufficient calcium is available. When we chelated calcium with BAPTA, we may have interfered with TRPM, but we may also have inadvertently made TRPC more available for activation by cholinergic signaling through the production of diacylglycerol, or one of the other mechanisms by which TRPC is activated. In figure 6, we are showing a simplified diagram of cholinergic signaling. Cholinergic signaling in CA1 pyramidal neurons is dominated by the muscarinic pathway, which activates phospholipase C and ultimately results in IP3-mediated release of calcium from the endoplasmic reticulum. This is the calcium source, which is thought to activate TRPM. There is some overlap with the TRPC pathway, as TRPC can be activated directly by diacylglycerol, which may explain our results in figure 5. An additional layer of crosstalk may be present as calcium entering through activated TRPC channels may be activating TRPM channels. This could also help explain some of our mixed results with SKF in figure 4C. It may come down to the crosstalk between TRPM and TRPC pathways. If IP3-mediated calcium released from stores activates TRPM channels, then blocking TRPC would do nothing, and the cholinergic-induced shift to the right would still happen, which is what we see here. If calcium entry through activated TRPC receptors is a major factor in activating TRPM channels, then blocking TRPC would indirectly block TRPM, and the cholinergic-induced shift to the right would be reversed, which is what we see here. If both mechanisms occur, but to different degrees in individual neurons, we may get mixed results when blocking TRPC, but consistent results when blocking TRPM. Finally, in order to investigate cholinergic modulation in a more nuanced fashion, we looked at dose-response effects of acetylcholine, the endogenous neuromodulator. In figure 7, you can see that the shift to the right is more subtle with acetylcholine than it is with carbachol, and it increases as the dose increases. We already see a significant shift in 2 micromolar acetylcholine and a further shift in 10 micromolar acetylcholine. These results suggest that the extracellular concentration of acetylcholine may be important in the modulation of firing rate responses within a cell's place field. To sum up, our results show that cholinergic modulation shifts the firing response of CA1 neurons to the right on the injected current ramp, and that TRPM4 channels appear to be at least partly responsible for the shift. It seems likely that variable levels of cholinergic activity contribute to shifts in the center of mass of place fields by modulating intrinsic ion channels. I would invite you to check out our companion poster that uses a model to explain some of these results. Thank you for your attention.